Welcome students. Today we are going to see the experiment observation of polytene chromosomes from chironomous larvae. So these are the chironomous larvae. So usually you can get this chironomous larvae from this aquariums. So usually they use this for fish feed and all. So these are also called as blood bombs. So mostly we will pick uh, the live larvae for doing our experiment. So we can take this larvae and from there we will dissect the salivary glands, and from those salivary glands we will stain and then we can observe the polytene chromosomes. So from here I am taking the live larvae and putting in 0.8% NaCl solution. So some larvae were picked and kept in 0.8% NaCl solution. So from here we will pick the larvae and we will dissect them. So I am taking the larvae and placing them on glass slide. So usually you can place the larvae and place 1 drop of NaCl. So since we have kept this in NaCl solution, so I have taken on the glass slide with a drop of NaCl solution, and this is a dissection microscope. So we are using this to dissect this larvae, to dissect the salivary glands from this larvae. So the glass slide was placed on dissection microscope. So you can focus from there. So this is the larvae. The dark portion there, it is the anterior portion, so their mouth parts will be there. So by that you can identify the anterior portion, and you have to pull out the salivary glands from the anterior part of the larva. So 2 needles were used, one needle to hold the larva and then another needle was used to pull out the mouth parts. So along with the mouth parts you will get the salivary glands also. So later you can separate the salivary glands and you will fix it in HCl solution, and then we can stain it with acetocaramine. So here I am pulling out the salivary glands. So the glands were pulled out like that. So here you can see the gut is also coming, and from the mouth parts we have to separate the salivary glands and this gut. So now you can see the separated mouth parts and the salivary glands, that light transparent structures you can see, those were salivary glands. So in this the staining and fixing is done. For that added 1 drop of acetocaramine stain, and then excess stain is removed by using the tissue paper. So after that the salivary glands were squashed, okay, and then you can view under the microscope. Here, okay, so now after staining the slide was focused, so you can see here the polytene chromosomes. You can see the banded structures. So these are the polytene chromosomes. So it appears like a banded structure. So not only from chironomous larvae you can isolate this, you can dissect the salivary glands from Drosophila larvae also. So these are the Drosophila larvae, third instar larvae. Here also you can remove the salivary glands and the same procedure is done for staining and all. So you can see this video for dissection of this Drosophila larvae. So the anterior portion, by using forceps, the anterior portion, the mouth parts were pulled out slowly, and you can see the mouth parts were pulled out slowly, and you can see two salivary glands. So since you have that black portion, that is called the mouth, that is the mouth parts, so those things has to be removed for staining these salivary glands. So the mouth parts are being removed. So very gently and slowly you have to do, otherwise you will disrupt the salivary gland. That should not be done. So these are the salivary glands. So here they are attached with some fat bodies. The dark portion is called fat bodies. So this is the picture showing the salivary glands.